So a couple things. One is I did get a review session scheduled. That is for Sunday at 4. And it will be in ALS 4001. And yes, I will videotape that. And yeah. So other things is I have, in fact, gone through all of your corrected exams and so forth. And I'll give those back at the end, and I'll say a little bit about them at the end so you'll understand what I've done on those, etc. I've also brought note cards, so make sure you have a note card because, as I said, you will have to bring a note card I give you in class to the thing, even if you don't fill it out. So make sure you bring a note card to class with you. And if you're watching in TV land and you're not getting a note card, then you'll need to come see me, of course, to get a note card so that you can bring it to the final. If you don't bring a note card to the final, you will lose points, and there's a reason I do that. So please do bring the note card to the final. And it has to be a note card that I give you, so make sure you get the note card from me. Okay? All right. And I don't do that to be mean. There is actually a reason. I won't say it here, but there is a reason I actually do that. So last time I started talking about nitrogen metabolism, and as you can see looking through it, there's a lot of material there for us to get through. So I'm actually going to be easy on you. Yes? How long is the final? So the final typically is about 150 points, which means it's about 1.5 times as long as the the exams that you've had so far. And it'll be the same format as the other ones, just more of each thing. Yes, Nick? That is ALS 4001 for the review session on Sunday. The final exam is in this room at 9:30 on Wednesday. So we get to see each other in the morning, see if we're actually alive in the mornings or not. I'm not. I am actually. I'm a morning person. So, okay. So let's, what I want to do today is, as I said, I want to go a little easier on you. There's a lot of material. So I'm going to go through some of the real highlights of the stuff that's there. I'm not going to go into a lot of depth with anything, at least hopefully not as much depth as I tend to go through and stuff. So let's do that. So first, I think I showed this last time, but if I didn't, I'll show you this. And I don't show you this to have anything for you to memorize or any of that, but when I talk about anaplerotic reactions, I talk about the sort of interplay between the things in the citric acid cycle and all these other processes that happen out there. So you can see how the amino acids play into all of those. You can see they either are broken down into citric acid cycle intermediates in many cases, or they're made from citric acid cycle intermediates in other cases. And so that's literally what anaplerotic means. But this tells us two things. One is that the amino acids are very tied to other metabolic pathways. And number two, that the citric acid cycle is, in fact, very central to all kinds of metabolism, which is one of the reasons we call it a central metabolic pathway. So that's just a quick glimpse of that. The thing I mentioned the other day being Wednesday, about the interplay between serine and glycine, and I said that there were molecules called folates that were important for that. And folates are very important in something we call one-carbon metabolism. One-carbon metabolism, okay? And one-carbon metabolism, as its name would suggest, involves a single carbon atom, okay? So there are several different folates, and I'll talk about another one near the end of the lecture today. There are different kinds of folates that have different kinds of carbons. So here's one that has a CH2 on here. We have others that have a CH3 and so forth. So they have slightly different names. And that's not really important for our purposes. What's important is that a very common way of making folates is in the metabolism of serine and glycine. Serine and glycine are very important in the synthesis of folates. Okay? And because we'll see folates are involved in nucleotide metabolism, it means that serine and glycine metabolism is also tied to nucleotide metabolism. Now, I'm not going to talk about sulfur per se, but I will remind you that Cysteine is an amino acid that contains sulfur. Methionine is the other amino acid that contains sulfur. And sulfur metabolism is, like nitrogen metabolism, a very important consideration in the environment. There are organisms that literally fix sulfur and allow us to ultimately take up sulfur that we can use in the form of amino acids. And that's pretty much all I want to say about that. The other sulfur-containing amino acid is methionine. And methionine actually is another very important compound when we talk about one-carbon metabolism. So I talked about folates being involved in one-carbon metabolism. What does it mean to say one-carbon metabolism? It means these folates, for example, can donate a one-carbon group to another molecule. That's what it means. Okay? So S-adenosylmethionine is a real good example of a molecule that can donate a methyl group. So, for example, in the synthesis of thymine, okay, we synthesize thymine from uracil. And the difference between thymine and uracil is a methyl group. That methyl group doesn't come from this particular molecule, but it comes from a folate, actually. But that's a one-carbon addition to a molecule. So that's what one-carbon metabolism is. So S-adenosylmethionine is also involved in one-carbon metabolism. And there is the carbon that's donated. There, right there, you see is the methionine part of the molecule. And over here is the adenosyl part of the molecule, meaning that it's got an adenine attached to it. Yeah, so I'll just leave that at that, I think. Okay. Last time I mentioned essential amino acids, and I didn't show you them. You don't need to memorize these or anything like that. But it reminds you that in human beings, about 10 of the 20 amino acids are essential. And as I said, the number count varies with what we talk about as far as the age of the human being. And it also varies from organism to organism. So if we're talking about dogs or cats, that might be slightly different than the essential amino acids for humans. Okay, now, catabolism of amino acids is another consideration. Okay? And it's from catabolism, that is the breakdown of amino acids, that we get the terms glucogenic, ketogenic listed. Okay? So when we list the amino acids, we list them according to whether or not they are metabolized in the form of glycolysis/gluconeogenesis intermediates. Those would be glucogenic, meaning that you could make glucose from the breakdown products of those amino acids. Or if they're ketogenic, that is, if they involve acetyl-CoA. Okay? So acetyl-CoA could, of course, be used to make ketone bodies. You see that there's a pretty large group that are glucogenic. There's a quite small group that's ketogenic, and then there's another 4 that are actually both. Okay? So that's the glutogenic and ketogenic. Nitrogen, as I mentioned last time, is in the form of ammonia or ammonium, is a toxic compound for cells. So organisms have to balance how much nitrogen that they're actually carrying around. And so all organisms have a means of excreting nitrogen so that they get that toxic ammonia out of their cells as appropriate. So there are different forms of nitrogen that exist, and these two are essentially the same, this being what happens when you dissolve ammonia in water. But these two forms here are, these ammonium/ammonia, I'll treat as the same thing. These two are one thing that can be excreted by an organism. Urea is what we excrete in our urine, and you can see there's 2 amines that are on there that readily go away. And uric acid, as I mentioned, is also a form of a molecule that's used for excretion. We will see that uric acid is actually made during the breakdown of purines. So purines are, of course, nucleotides or bases of nucleotides like adenine or guanine. And so their breakdown leads to uric acid. it is related to gout, and I'm going to say something about that as well. Okay? Okay. Well, actually, maybe I will go ahead and say something about gout right here. So uric acid is produced in our bodies as well. We don't use it as a primary means of excreting nitrogen compounds, although we can excrete uric acid. The problem with there is a problem associated with uric acid, though. You can see uric acid can lead to urea, which is, of course, the form that we excrete nitrogen in the form of. But uric acid is problematic. So how do we get uric acid? I said we break down purines. And during the breakdown of purines, uric acid is an intermediate in that process. A problem arises if you have too much uric acid in your body, and it's an odd problem. The problem is that uric acid is not very water soluble. So when it's produced, if it's produced in sufficient quantities, and in some people it doesn't take very, very big quantities, what will happen is it will precipitate out of the aqueous solution and it will crystallize. And where it will crystallize is the place where the uric acid concentration is the highest, and that's the farthest down in your body due to gravity. So the crystals of uric acid will accumulate in your feet. So there's a condition known as gout that arises. It tends to happen in older people, although it's not exclusive to older people. And it's first diagnosed as an extremely painful big toe. Because that's where the crystals actually form. What's happening with the crystals is that they are impacting a nerve cell. And that nerve cell, it can be quite excruciating pain. It used to be depicted in the movies very comically. Oh, the person would be holding their toe and screaming and hollering and so on and so forth. There are treatments now that alleviate the production of uric acid and therefore bypass the whole pathway in general. But one of the interesting things that's been found about gout is that people who have gout tend to have a lower incidence of multiple sclerosis. And it's not known why that's the case. It's thought that there may be some antioxidant property of uric acid that helps prevent the disease. Yes, is that a question? Okay, so iodide, to my knowledge, has no link to that. I don't know that there is one. I'm not saying there's not. I'm just not aware of one. Iodide, of course, is a factor in thyroid gland, so it may have, if it's related to that, I would guess. I'm not familiar with anything like that. One of the things they used to do in the old days was they called gout rich man's disease, because the only people who ever got it were those that ate diets that were rich in red meat, for example. Because those tended to be heavier in purines. So the more purines you're getting, the more likely you would be to contract gout. Seafood, I just don't know. Sorry. Yes? I'm not aware of any alcohol effect on the production of uric acid, but again, I'm not a medical person, so I just can't tell you. Don't know. Okay. All right. So that's the uric acid story, kind of an interesting one. And when we talk about urea, it's appropriate that we should talk about the urea cycle. Now, this is not a pathway I'm going to give you to learn, so don't worry about that. I want you to be aware of what it is. So the urea cycle is a cycle for managing nitrogen. It's a cycle for managing nitrogen. There are a couple of molecules in it that I think that you should know. Okay? Now, I'm not going to have you draw a pathway, or don't do that. Okay? But there's a couple of molecules that are interesting. One is right here. It's the amino acid arginine. Arginine is the precursor of urea. So if you start here, for example, with arginine, and you clip off the end of the molecule right here, what you end up with is urea. And then the other molecule that you end up with is ornithine. Okay? Ornithine is an amino acid. It's an amino acid that never makes it into protein. Okay? Both ornithine and citrulline, that you can see in here, are amino acids, but they're not used to make proteins. You do see these two commonly in what people describe as energy drinks. Okay? energy drinks. They're very common components of energy drinks, and some people think that not only do they give you more energy, but they give you more brain power, et cetera, et cetera. Personally, I've never seen any evidence for that, not from personal experience from taking energy drinks, which I won't take, but I've never seen any scientific evidence for either claim. However, that claim is out there. As I look at nitrogen metabolism, I would probably not recommend messing with it. So my advice would be don't go out be getting ornithine and citrulline supplements and so forth, because you might be mucking with something that you really don't want to muck with. One of the things that happens with this cycle is that people who have deficiencies of any of the enzymes in that cycle have some very severe neurological problems because they can't properly get rid of nitrogen. And that, again, toxicity of the ammonia that's there is real problematic for them. Okay, so those are the molecules I want to talk about: ornithine, citrulline, and arginine that are involved in the urea cycle. This, you might wonder what this is here. This is the mitochondrion. This is the cytoplasm. So you see a couple reactions happening in the mitochondrion. You see the rest of the reactions happening in the cytoplasm. Okay. So that's what I want to say about nitrogen metabolism per se. A tangent of nitrogen metabolism is the metabolism of nucleotides. So that's going to be our very last topic, and I want to just spend a little bit of time on that. First, I'll talk about purines. Purines, you remember, include adenine and guanine, or adenosine and guanosine, if we're talking about nucleosides. The bases of these look like this, and you've seen these before. I'm not going to, again, you're not going to memorize that structure. But the reason I show this figure is to impress upon you how easy it is to make a base for a nucleotide. It's very easy. Very simple starting materials. This part comes from glycine. This part comes from one carbon metabolism of a folate. This comes from glutamine. This comes from another folate. This comes from aspartic acid. And this comes from carbon dioxide. It's not difficult for a cell or even a non-cell to make a base. Okay? When we look in meteorites that fall from space, we've actually seen evidence of the chemistry that gives rise to this. Okay? So we know that these kinds of reactions can and in fact do occur independent of living organisms. So our idea of the chemical basis of life is a very solid one. And it's interesting also to see that the things that are the most important molecules in us in terms of information are actually made from things that we know are abundant all around. Now, there are a lot of steps in both purine and pyrimidine nucleotide metabolism, and we don't need to look at all those to understand them. In the synthesis reactions of purines, there are 10 steps, and I'm only showing you a part of the process here. The 10 steps start with very simple compounds, as I described to you, and these simple compounds give rise to a molecule known as IMP, inosine monophosphate. Okay? I'll close that door. Excuse me. Now, I mentioned IMP because it is the branch point between the synthesis of AMP and the synthesis of GMP. AMP being adenosine monophosphate, GMP being guanosine monophosphate. Okay? This pathway and this pathway, each one is interesting. If we look at the pathway going down to AMP, we see, first of all, that it uses energy from GTP to make it. And at first you might say, so what? Well, if you think about it, the reason the cell is going down this pathway is it doesn't have enough adenosine nucleotides to make things. So it wouldn't make sense to use ATP to make ATP because it's already short. So presumably GTP is present in greater abundance. Okay? If we look at the synthesis of GMP from IMP, we see that ATP is used as the energy source for it. Again, no surprise, because we're making guanosine nucleotides because they're in short supply, and presumably the ATP is in greater abundance. So that's an important consideration for both of those. Now, one other thing that I'll point out is that GMP feedback inhibits this enzyme. So it feedback inhibits its own synthesis, and AMP feedback inhibits its own synthesis. Okay? Now, I've mentioned briefly before that nucleotide metabolism is one area of metabolism where balance is really important. You don't want to get too much of one nucleotide compared to others. I talked about that with ATCase, and we're going to see that in a couple of minutes again. And we don't want to have too much of anything. So if we start seeing one we're getting too much of, we will shift synthesis to the other. And if we start to get too many pyrimidines, for example, we will start to shift synthesis to purines. If we start making too many purines, we'll start shifting synthesis to pyrimidines. So there's very, very good balance that happens with all of the nucleotides. Yes? No. AMP, these are both purines because they have the double ring. So we're talking about purine metabolism. Okay. Well, that's the synthesis. I've already talked a little bit about the catabolism, and I'm not going to go back through that, but I will tell you that the uric acid is a big consideration in the breakdown of that. If you want to have a headache, I guess you can look at that and figure out regulation, which I'm not going to hold you responsible for. But I will mention one enzyme that's named that you should know and with respect to regulation. It's the very first enzyme in the pathway, and it's called PRPP aminotransferase. I'm sorry, PRPP amidotransferase, AMIDO. Okay. That's the most important regulatory enzyme for the synthesis of purines. And it's interesting how it's regulated. Okay. The interesting thing is that the enzyme is completely inhibited only when it's bound by both ATP and GTP. ATP and GTP. If it's only bound by one, it slows down, but it doesn't stop. And that allows the production of the other enzyme. So the other molecules. So if ATP is abundant and GTP is not, if you completely turn off the enzyme, you wouldn't be able to make GTP. But by keeping the enzyme slightly active, all that can be directed into making GTP. So it's kind of a cool setup that's used to control the purines. Now, in addition to the pathways that make nucleotides from scratch, there are also pathways that make nucleotides from existing pieces. So we think about a nucleotide. What's it got? It's got a base, it's got a sugar, it's got a phosphate, right? At least one phosphate. So what if I break off the base, and then all of a sudden I decide, oh, wow, I need to make some purines. Do I have to go back and start from scratch, or can I use that base to remake purines? Well, you can. And that's called salvage, like a salvage yard that you can pull pieces off of an old car to use in your car that are cheaper than going out and buying new parts. So too is salvage nucleotide synthesis important for cells because it allows them to remake nucleotides without wasting the energy of having to start from square one. And so salvage synthesis of nucleotides is really important. I tell you this because there's an interesting syndrome called Lesch-Nyhan syndrome. And it involves this enzyme. We're not going to worry about the reactions, but it involves this enzyme called HGPRT. HGPRT catalyzes what seems to be a relatively minor reaction for cells. It occurs in a couple of ways, and you can see it going on right here. Don't worry too much about the details. But what's interesting about this enzyme is when it's deficient, even though this appears to be a fairly minor reaction of cells that occurs inside of cells, the disease associated with it is horrendous. It's called Lesch-Nyhan syndrome. It's written on the slide. It's LESCH-NYHAN. And people who have this disease have severe mental retardation typically, and they will eat their own lips and fingers. And it's not known why or exactly what the neurological disorder is with that, but they have to be restrained because they will literally eat that off. And one of the things that they can eat when they're restrained are actually their lips, and they will eat them. It's yeah. It always gives me the creeps when I think about that. But they eat them off, so yeah. Okay, that's purines. We're getting through there, guys. Let's talk about pyrimidines. Pyrimidines are a little different from purines. We start making, well, actually, no, I won't talk about that. Pyrimidines also have very simple compounds that go together to make the base in a purine. And the base in a purine is a single ring, okay? A single ring. This is an early pyrimidine that is being made. And we can see that it leads to production of UMP. That's the first pyrimidine that's actually made in pyrimidine synthesis. This is a shorthand abbreviation. We can see the ring structure here, and we can see the ribose with the phosphate down here. So they made these small so that you could look more closely at the ring structure there. So UMP is made by this pathway, and the enzyme that starts this pathway is one that you've seen before. There's ATCase. And ATCase, you remember, helped the cell to balance between purines and pyrimidines. If we had high levels of ATP, which was a purine, then that meant that purines were greater than pyrimidines, so this enzyme was activated. And when pyrimidine in the form of CTP was in high concentration, then this enzyme was inhibited, which meant that the pyrimidines were in higher concentration, and the cell wanted to tamp those down. So the cell balances both the purine levels and the pyrimidine levels, partly through the allosteric regulation of ATCase. Now, the UMP is converted into UDP by an enzyme called nucleotide kinase. Okay? This is a nucleotide monophosphate kinase. Nucleotide monophosphate kinase. This guy is a nucleotide diphosphate kinase, and this one is one we're going to focus on. Okay? And there's a reason why. Every monophosphate has its own nucleotide monophosphate kinase. UMP has one specific for it. CMP has one specific for it. AMP has one specific for it. And GMP has one specific for it. When we go to the diphosphate, however, all of the diphosphates are converted into triphosphates using this kinase, the nucleotide diphosphate kinase, or as you call it, NDPK. So NDPK converts all of the diphosphates to all of the triphosphates. That's true whether they are pyrimidines, that's true whether they are purines, and that's also true if they're deoxyribonucleotides, and we'll talk about them. We're talking about ribonucleotides so far. We haven't talked about deoxyribonucleotides. Okay? So NDPK converts all diphosphates to all triphosphates. And you can see the energy is ATP. This guy here can use a variety of energy sources. This one could use GTP, or it could even use CTP or UTP as necessary. So as I said, UTP is the first nucleotide that is made, and CTP is made from UTP. So CTP really is the end point of the pyrimidine synthesis pathway. It uses this enzyme called CTP synthetase, and this enzyme is inhibited by CTP. So now if CTP gets too high, it will not only inhibit the ATCase, but it will also inhibit this synthesis so that this can build up and there can be balance again. So we see really amazing balances that happen in the synthesis of nucleotides. And that's the regulation which I talked about. I'm not going to say anything more on that. Catabolism we're not going to worry about. Pyrimidine catabolism is not nearly as interesting as purine catabolism. And all I will tell you about it is that it just leads to urea, ultimately. Leads to urea. Now, the last things I want to talk about are the deoxyribonucleotides. Everything I've said so far here in purine metabolism and in pyrimidine metabolism is describing the ribonucleotides, the ones used to make RNA. The deoxyribonucleotides are synthesized in an interesting fashion, okay? A very interesting fashion. This depicts the synthesis in a very complicated way of the deoxyribonucleotides. That's not the one I want to show you. Oh, did I pull that off of here? I did it again. All right. Okay. Well, the important reaction is actually right here. Okay. So you can ignore everything else on this slide except for this going to this. All right. The substrate for ribonucleotide reductase is a ribonucleoside diphosphate. What is a ribonucleoside diphosphate? ADP, GDP, CDP, UDP. Okay? So any of the diphosphates that are ribonucleotides are substrates for ribonucleotide reductase. Now, this is the enzyme that makes the deoxyribonucleotides. Okay? It makes the deoxyribonucleotides. ADP gets converted into DADP, GDP into DGDP, CDP into DCDP, and UDP into DUDP. So it's the diphosphates that are there. And again, we see one enzyme doing all of them. There's ribonucleotide reductase that's handling all of those. Okay? So when you're getting to learn pathways, this is a great time saver, just like NDPK is a great time saver in terms of memory, because you only have to learn one enzyme to learn what it's doing. When we get them to the diphosphate, if we want to build them into nucleotides, into DNA, we need to convert them into the triphosphate, and that is done by our friend NDPK, because NDPK converts the diphosphate into the triphosphate. And I'll explain this multiple step here thing in a second. But you can see the diphosphate to the triphosphate, the diphosphate to the triphosphate. And so NDPK does all of those, just like it did all of the ribonucleoside diphosphates as well. As a result of this, the cell then has the triphosphates necessary for making DNA, in this case, or RNA if we're talking about the ribonucleotides. Now, the one hook is thymidine. So you notice I showed you how to make uridine-containing nucleotides, that is those that have uracil in them, but I haven't shown you thymidine. And that's because thymidine, which is the T of DNA, is made in an unusual way. It's made from UDP. So UDP is the starting material to make thymidine nucleotides. And by the way, some people just say thymidine. They don't say deoxythymidine. I will say deoxythymidine because I think it just alleviates confusion of students. The reason people say thymidine is thymidine doesn't appear in RNA, so therefore it's automatically deoxy. And it's like, well, duh, let's just call it deoxy to keep it like consistent with everything else. In any event, the deoxythymidine is made from DUDP. Now I'm going to take you through the odd steps that it goes through, that the cell goes through to do this, and tell you why. So we start with UDP, which ribonucleotide reductase converts to DUDP. Okay? DUDP is then converted into DUMP, dump. Right? I like dump. Everybody always likes dump, right? What in the world is the cell doing? It's converting it into dump, all right? And the reason it's converting it into dump is so that it can make this guy right here, DTMP. Okay? DTMP is the thymidine nucleotide, all right? And the enzyme that does this catalysis is called thymidine synthetase. And yes, that's an important enzyme, thymidine synthetase. And this is the enzyme that's using folate. So you see methylene coming off. That's the FH2 over here. Methylene is getting put onto there in the form of making a methyl group. Notice there's no methyl group here. Notice there is a methyl group here. Here's some one-carbon metabolism happening. Okay? DTMP is phosphorylated by a nucleotide monophosphate kinase. And then our friend NDPK converts it into DTTP. Okay? Now, I want to say one more thing, and I'm actually going to finish a fair amount early today. I'm going to say one more thing about this molecule that's important because it actually has implications for cancer. That's the last slide. Okay. Now, here's the reaction that I described to you of DUMP going to DTMP. And here's this giant molecule which has this single thing here that's ending up here. Okay? It starts out as a CH2. It goes up here as a CH3. That causes this guy to not only lose a carbon but also to get oxidized. In order to get back to this state, okay, there's a couple of reactions that have to happen. And just like you've seen with NAD and NADH and other things, the cell has a limited amount of folates. So if it doesn't do this conversion back here, what's going to happen is everything is going to end up as this, and this reaction is not going to be possible. Does that make sense? So this recycling of the folates is very critical for cells. They're doing it all the time. So why do I tell you that? Well, if it's critical and we're making a nucleotide, what happens if we block this thing? What's going to happen to DNA synthesis? Right? Well, there are times we might want to do that. A real good time we might want to do that is if we have a cancer cell. A cancer cell is rapidly growing, rapidly making DNA. And so if we treat a cancer cell with something that blocks this process from happening, okay, then this cancer cell will run out of DNA, nucleotides, and it will die. Now, the downside is that our healthy cells are going to be suffering in the process, and this will only work if the cancer cell is dividing more rapidly, and it dies first. So one of the chemotherapy drugs that we give to patients is called methotrexate, and it's given for other reasons than chemotherapy as well. But in higher doses, it's given in chemotherapy, and the idea is it inhibits this enzyme right here, dihydrofolate reductase, known as DHFR. It inhibits this enzyme so that this entire recycling process can't happen. The idea with the chemotherapy of this is that you give it to a patient for a period of time, and then you withdraw it so that in the time that it's in the patient, it's hopefully killing more cancer cells than it's hurting or killing living cells. But it does kill some living cells, and that's one of the reasons why some cancer patients have very difficult chemotherapy. They may get very ill, lose their hair, et cetera, et cetera. And that's partly because some of these drugs will do exactly what this guy does, which is harm healthy cells. Yes? It's called methotrexate, methotrexate. Okay? Methotrexate. And you might say, well, how does it work? And I'll say it's a competitive inhibitor. What would that tell you? Thinking back to our earlier part of the term, what does a competitive inhibitor do? It competes for the binding site for what? For the enzyme, which means it must have what property? It's similar, and it's actually similar to this guy right here. It looks very much like this guy right here. Okay? So that's what methotrexate does. Okay, believe it or not, I actually, after many promises, I actually have finished early. So I want to take a couple of minutes and, first of all, tell you about the exams I'm getting ready to hand back to you. I also have a note card to hand out to everybody, and it's one per person. So I will give those to you in just a second. And before I do that, let me tell you a little bit about the exam. So I went through, and for all of you who turned in regrades, I regraded every one, and I looked to see, did the answers that you gave me, were they right or not? So not all the answers that people gave me were right, and that I understand that. But for every one you got right, I made a little note on the side. You'll see my numbers there indicating if you got full credit or if you'll see partial credit. I wrote the numbers in parentheses, so you'll see the numbers beside there. Then I tallied up all those numbers, and on the front of your exam, you're going to see a number that's got the sum of all those individual numbers, right? So maybe you got, 30 points, okay, on the regrade. And you'll see on the front sheet, in parentheses, you'll see 30 points. That make sense? Then I decided what percentage of those points would people get back. Okay? And so what I decided on was to give everybody 30% of their correct points back. So you'll see that. I wrote that on your exam. You'll see your original score. Then I'll say plus 4.9 or whatever it worked out to be. And then I'll have a final number equals something percent. And that last number is the grade I have recorded for you that will be used to calculate your grade. Does that make sense? Because you're going to see several numbers on the front sheet when I hand this back to you. All right? So make sure, if you have any questions, come see me. But when you see that number with the percent, that's the number that I'll use to calculate your final grade. Okay? Make sense? All right. So I will say, first of all, that was a very good exercise. I learned a lot from talking to you, and I very much appreciate the feedback that you gave me for that. It was a very good exercise to do, and I very much appreciate you taking the time. I hope for all of you that was also a learning experience for you. It certainly was for me, and it gave me a chance to get to know some of you a little bit better, which was also very nice. You've been a very good class to work with. I've been very impressed with the attendance and the seriousness with which you've all taken the class, and that has all been very, very good. So I thank you very much for that. So what I'm going to do first is pass out note cards.